If you take those plastics and you heat them above about 70 degrees centigrade, so not boiling, but near, so 93 is again boiling in Yellowstone, those plastics start to fall apart. Essentially, what you're doing is you're unwinding the polymers that form that plastic. Well, if we can find a microbe that can grow at 70 degrees, that can hydrolyze those plastics and turn them back into their monomers, we've just solved a huge problem.
Thanks for joining us again on the Voices of Greater Yellowstone podcast, where we share the stories and science of the Greater Yellowstone ecosystem. I'm your host, Emmy Reed, and welcome back to part 2 of our Thermophiles in Yellowstone conversation. If you haven't turned into part 1 first, I recommend pausing this episode and hopping into the previous one to learn the basics about the terrific thermophiles. I'm excited to continue our conversation with Dr. Eric Boyd and dive deeper into the world of thermophiles. Did you know that the thermophile species discovered in Yellowstone has been used in important scientific discoveries related to crime scene investigation? If you're an avid true crime podcast listener, you'll definitely want to stick around to learn about that discovery and application. We'll also cover how thermophiles played a role during the COVID-19 pandemic, what could still be waiting to be discovered in Yellowstone's thermal features, and we'll also get into some great listener questions submitted by our podcast insiders. All right, let's continue this burning conversation about thermophiles. Gotcha. We'll give it a few million years and see how it looks then.
There you go.
If a thermophile is outside of its ideal temperature range, what happens to it? Does it die? Or how long can it live outside its ideal temperature range?
Interesting. So there's kind of two different ways to think about this. You can go too hot, and that's not a good place to be for any form of life. And so I'm going to go back to this example that we were talking about with the egg. And you cooked that egg, you made it too hot, and those proteins permanently were denatured. That's why they went from liquid to solid. That you're denaturing the proteins, the albumin proteins in that egg. Now, the same thing will happen to a thermophile. They may be optimized to grow at, say, 200 degrees Fahrenheit. But if you put that organism at, say, 210 degrees Fahrenheit, you're going to denature proteins. You're going to denature those lipids. You're going to start to melt their cell walls, their cell membranes. And so going too hot is never a good place to be for any form of life. Now, if you go too low, go the other direction. So I take that thermophile that's, say, optimized to grow at 200 degrees Fahrenheit, and I put it at 100 degrees Fahrenheit, it'll just go dormant.
Oh.
That won't kill it.
Okay.
And in fact, one of the ways that we preserve microbes is we freeze them. And it's the same reason when you want to preserve food, put it in the refrigerator, cool things down. Put it in the freezer, it'll last a lot longer, type of thing. But that doesn't necessarily mean that you killed the organisms.
Right.
Right? If you had frozen ground hamburger and you thawed it overnight in your refrigerator, that would be fine. And you could make your burgers just like that. But if you left it on your countertop for a week, you would not want to eat that burger. Freezing did not kill any of the microbes that were present in that burger, if that makes sense.
Total sense. That's a really good way to think about it. Thank you. So thermophiles are obviously incredible organisms that have played roles in some important scientific advancements. First, I was wondering if you could talk about why NASA funds the study of thermophiles and other extremophiles.
For sure. We've kind of touched upon this a little bit, but essentially, Earth is a special place where the majority of it, if you think about the United States, it's a pretty habitable, if you will, country, right? You said that you didn't like before we got talking here that you don't like humidity. Well, I don't either,
Ooh.
but still, even humid climates are fairly habitable. Okay, but that's not the case for the majority of planets in our solar system or beyond. There's very few places where you've got nice ambient, if you will, temperature or pH or salinity. The majority of the environments that you find um, outside of Earth are extreme. And because of that, if you're going to find life on other environments, which is what NASA's one of NASA's charges is to search for life in the universe. Um, the likelihood is that you're gonna if you're gonna find it, it's gonna be in a location that is not 37 degrees C, pH circumneutral, fresh water. It's gonna be salty or briny. It's going to be cold or hot. It's going to be acidic pH or alkaline pH. It's not going to be what we would term like kind of the Goldilocks zone. And so NASA then therefore needs to understand what to look for in these extreme environments. And you can provide insight into what kind of organisms or what kind of processes support life in these environments by studying these extreme environments here on Earth. We've already talked about understanding the origin of life on Earth. If you understand what conditions led to the origin of life on Earth, you can also look for such conditions on other planets. But you might also be looking at extreme conditions. So, for example, Europa. So, Europa, a moon of Jupiter, um, very oxidizing conditions on the surface, a surface ice layer, a rocky interior. Looks like that you could generate a lot of chemical reductant when you react water with those rocks. You've got similar conditions to what you might expect in a briny ocean between that rocky interior and that icy layer. Can we identify environments that might have similar brine compositions, salt compositions, if you will, and ask the question whether or not there's life there in those environments? What kind of biosignatures do they produce? Do they produce diagnostic gases or diagnostic lipids that we could potentially search for? What kind of processes are supporting those organisms? Okay, and those are the kind of analog studies that we'll do here on Earth to inform the search for life elsewhere. And you say, okay, well, why is NASA ever going to search for life on Europa? Well, they are. There's a mission called the Europa Clipper um, that's on its way now to study to look for evidence for life on Europa. Pretty neat.
Very neat. Um, So one of the most I would say famous, seems like it's pretty famous in the microbiology world, discovery of a thermophilic species was Thermus aquaticus in Yellowstone National Park. And so could you tell me a little bit about that discovery and how its discovery has led to better crime scene investigation and biomedical research?
Yeah, so Thermus aquaticus. So we've already mentioned his name once, Tom Brock, and his graduate student, Hugh Freeze, were the first to isolate Thermus aquaticus. They isolated it out of Mushroom Pool, which is in the Middle Geyser Basin, off the boardwalk, so you can't just go there and see it. But there's plenty of pictures online if you want to see what Mushroom Pool looks like. There's nothing spectacular about it or unique about it. It's very similar to other circumneutral alkaline springs in that area. Isolated Thermus aquaticus, and that was one of the first thermophiles ever grown. They themselves weren't the ones who envisioned the biotechnological applications of Thermus aquaticus. So, all cells have biochemical machines to replicate your DNA. So you have these same enzymes because every time your cells divide and you grow, you need to copy your DNA, your chromosomes, or your genome. Okay, microbes are no different. Okay, but your enzymes are optimized to function to replicate that DNA at 37 degrees C. That's your body temperature. plus or minus. Well, Thermus aquaticus is optimized to grow at a much higher temperature, and therefore all of its enzymes are optimized to function at those higher temperatures. Make sense?
Yep.
Okay, and so now we'll turn to the laboratory environment. People envision this the need to amplify or to replicate DNA. They needed to do this for various molecular biology applications, and it wasn't easy to do. Um, they needed an enzyme for a chemical reaction or a biochemical reaction called the polymerase chain reaction that was thermostable. And so essentially, you run these cycles of repeated heating of the DNA where you unzip the double stranded DNA into single strands. And then you have this enzyme that then, with that single stranded DNA, replicates it and adds the same complementary base pair for that DNA. Okay, that's DNA replication. And then you have to heat it back up to separate those strands once again for the next round of DNA synthesis, if you will. Each time that you had to do this in this polymerase chain reaction, you had to add more enzyme because the enzymes that they were using were not thermostable. So every time that they heated those up to replicate the DNA, separate the DNA and replicate the DNA, they would denature those proteins that the polymerases that copy that DNA. So they needed a thermostable enzyme. Well, that's where Thermus aquaticus came in. And that's when biochemists isolated the DNA polymerase from Thermus aquaticus, Taq polymerase, and now they could run these instead of manually running these cycles, adding polymerase 35 times in 35 different cycles. You could just set up the reaction one time. And now what you're doing each time you run one of those cycles of PCR, if you will, you take 2 strands of DNA, you make 4. Take 4 strands of DNA in your next cycle, you make 8. Okay, so it's exponential increase in the copy number of those DNAs. And so you ask the question, how did this biochemical revolution, if you will, impact crime scene investigations and forensics? Well, oftentimes criminals don't want to leave behind evidence of their activities, right? And so they might just leave a skin cell or a hair with an attached skin follicle. Well, there's not enough DNA there to sequence. And so you can't fingerprint the suspect or the criminal with that amount of DNA. You need to increase the amount of DNA so that you can sequence it and then look for a match in a database, so on and so forth. Well, that's where PCR comes in. You can come in now and take those few cells with their genomes, you can amplify them millions of times so you have enough material to sequence and then therefore make a match to your suspect.
I just that's such to like go from a hot spring in Yellowstone to solving crimes across the country is quite the path for a little thermophile to take.
Absolutely.
Wow.
A multi-billion-dollar business from one organism, one protein from one organism out of one hot spring in Yellowstone. And we already talked about how biodiverse Yellowstone is. There's a lot of biotechnological potential in these thermophiles, no question about that.
Yeah, and like speaking of more potential, could you tell us how Yellowstone thermophiles played a role in the COVID-19 epidemic?
Well, there's a lot the same reason. And so again, so Taq polymerase was what was originally used in PCR reactions, and they've done a lot of tinkering with that polymerase. They've isolated other polymerases from other thermophiles that are actually even more. They're better. They are better at copying. The accuracy of them copying DNA is better than what Taq was able to achieve. Um, but it was largely the same question. So, are you COVID positive or are you not? And so we all remember having those nasal swabs,
We do.
and then they would go back into their fancy laboratories and essentially run a modified PCR reaction. And they would quickly know within a matter of literally less than an hour by running a modified PCR reaction whether or not you were COVID positive or negative. They were simply looking for sequences of that virus in that nasal swab from your nose using PCR.
Wow.
Pretty neat.
That's incredible. Does the park get compensated for these discoveries, or is there any uh not even like a relationship there, but yeah, how does the park play a role in this?
But the park, I mean, let's face it, the park played the critical role in this because they were the host. They're charged with preserving Yellowstone for future generations. So had the Park Service not been doing its job, there's a potential that vandalism, so on and so forth, could have led to destruction of some of these thermal features. And Thermus aquaticus was never discovered. Tac polymerase was never discovered, so on and so forth. It turns out that the Park Service, and by the Park Service I mean the American taxpayer, never received a dime in royalties from Thermus aquaticus, and that's because they didn't have the legal documents in place when they were issuing permits to do research in the park back when people were doing research in the park. Now, when I get a research permit from Yellowstone, there are legal legalities of what we can and cannot do with the materials that are collected. And I should mention we collect a very small amount of material from the park in a given year. But we have to enter. If we commercialize something, the Park Service gets a hefty share of any profit that's made between from that commercialization here at, say, MSU, Montana State University, and the Park Service. So we have an agreement, and we have to sign all of that away before we're ever allowed to collect samples, so that something like that doesn't happen again.
Okay. Yeah, gotcha. So the park lawyered up. That's good. Um, we talked a little bit about this, about just the possibilities of what could still be discovered when it comes to thermophiles in Yellowstone National Park. And I mean, could you even would you be comfortable speculating about what could still be discovered and what role it could play?
I think the sky's the limit.
Yeah.
And one of the things I like to always point out to people is humans are really good at creating problems for ourselves to deal with. And let's think about plastics, right? We make these materials, plastic for one of them, that aren't the reason plastic is so useful and why it's proliferated in society is because it doesn't break down, right? You can reuse it, it doesn't break down, so on and so forth. Well, that's great from a use perspective. It's not so great from an environmental perspective. Now we've got this problem with plastics everywhere. One of the neat things, and I know a lot of people are looking at this right now, is to use thermophilic microbes to degrade plastic. And he'd say, well, that seems like an odd application, but if you've ever taken, they're so the plastics that are recyclable tend to be plastics number one and two. So these tend to be your single use plastics, so which are the kind of the worst of all of the plastics. We shouldn't be using plastics and just throwing them away, but nonetheless, that exists. Um, if you take those plastics and you heat them above about 70 degrees centigrade, so not boiling, but near, so 93 is again boiling in Yellowstone, those plastics start to fall apart. And that's essentially what you're doing is you're unwinding the polymers that form that plastic. Well, if we can find a microbe that can grow at 70 degrees that can hydrolyze those plastics and turn them back into their monomers, we've just solved a huge problem, which is we can now recycle those plastics. So you make those plastics much more available for microbial activity to work on once you start to unwind them at 70 degrees, if that makes sense. Um, so that's one application. I know there's other applications where people are looking at some of the various pigments that thermophilic microbes make, trying to understand the biosynthetic pathways for how those pigments are made. In particular, blue pigments. Blue pigments are really hard to make industrially, and they oftentimes require really nasty chemicals to make. Well, what if you could make a blue pigment naturally inspired by one of these thermophilic microbes, and remove all of that hazardous, nasty chemical synthesis from the process? And so there's a lot of different applications, and I think honestly, it's the sky's the limit. If you want to think about how to make a process either happen faster or more cleanly, or to involve fewer toxic chemicals, then maybe there's a solution for that in a microbe from Yellowstone.
That's so cool. I'll be really excited to see how the next couple years to many years play out in this field. You're right. It really does seem like the sky is the limit to how thermophiles can play a role in our lives. Um, what's something maybe just unexpected that you've learned about thermophiles during your studies?
Interesting question. So we just published, yeah. I mean, there's been so many unexpected things that
Yeah.
we've learned, and that's essentially what forms the essence of a scientific publication. You want to tell the world something unexpected that you learned from your studies. We recently had this we published a paper on this organism, and very different from the paradigm for how we think the energy metabolism of a cell should work. Okay, and so you got up this morning, you ate your breakfast, and let's just pretend you had a banana. You had a really healthy breakfast,
All right.
and so you had a bunch of sugar for breakfast. That's essentially what a banana is. And now your body is taking that banana, it's digested it, but it's essentially your blood is circulating that sugar, and you're taking deep breaths of oxygen, and you're exhaling CO2. So essentially, you're breathing oxygen so that you can burn your cells can burn that glucose, that sugar. Okay, that's your metabolism in a nutshell. And thermophilic microbes will do the same thing, right? They will take essentially an electron donor, which in your case is that glucose, and they'll react it with an electron acceptor, which in your case is oxygen. And essentially, what they're doing is they're releasing the energy from that process, and you're using that energy to power your cells, to power your body. Microbes will do the same thing. Now, we discovered an organism that was essentially doing just that. It was breathing, in this case, it was breathing hydrogen gas. Right? Imagine that, an organism that's instead of eating glucose, it's eating hydrogen, rocket fuel,
Yeah.
and it's reacting it with oxygen, which is that's cool, right? I mean,
Yeah.
that's what NASA uses to launch stuff into space. That reaction right there. Okay, and so it's doing that. And what we found is that when we gave it sulfur, so elemental sulfur, that's that yellow stuff that you see scattered around all of these hot springs. Okay. Um, when we gave it sulfur, it would simultaneously also breathe that. And so essentially, you have what we call an aerobic metabolism. When you breathe oxygen, that's an aerobic metabolism. When you breathe something other than oxygen, anything other than oxygen, it's called an anaerobic metabolism. This organism, and so there's nothing unusual, honestly, about either one of those, an aerobic or an anaerobic metabolism. What was unusual and unique and super interesting was that this organism was doing both of those processes at the same time. And that's not supposed to happen.
No.
That's not supposed to happen. So it's not uncommon for an organism to grow aerobically and then all the oxygen runs out, and they'll switch to an anaerobic metabolism. In fact, your cells do that too. So like when you're running, you're sprinting, you're doing a 400-meter dash, and that last 100 meters, your muscles are just, oh, they feel like they're burning. That's because your body can't supply enough oxygen to your muscle cells fast enough. They're actually turning to a fermentative reaction where instead of burning glucose with oxygen, they're fermenting glucose. And one of the products of that is lactic acid. That's what causes that burning sensation.
Right.
Okay. But your body doesn't do both of those processes at the same time. They use oxygen until there is no oxygen, and then they ferment. What I just described is an organism that's doing both that aerobic and anaerobic process at the same time, which has never been described before, as far as we're concerned, for a beneficial reason at least.
Gotcha. Wow. I just like I understand, but like my brain just cannot wrap my head around that it can be both aerobic and anaerobic at the same time.
And I think the one of the things that one of my mentors always told me was to, when somebody tells you something can't be,
Mm-hmm.
right, this is the way it is, this is the paradigm, then look for an alternative. Because in the microbial world, and we've talked about just how many different types of microbes there are, there's almost always an exception to the rule, right? And this is an exception to the rule, right? Microbes should not, or life should not, simultaneously aerobically respire and anaerobically respire. That's not supposed to happen. Well, hey, it looks like it does happen.
Right. That's good advice. Good advice. You mind if we hop into some listener questions?
That'd be great.
All right. London from Montana asks, "What's your favorite thermophile?"
Huh. Uh, Great question. Anything that grows with sulfur.
Okay.
And I say that I did my PhD on sulfur respiration. So elemental sulfur, that again, that yellow mineral that you see all over hot springs, if you oxidize it, you're contributing to the acidification of a hot spring. And so those are organisms like sulfolobus, if you will. Those are archaea. If you reduce it, you breathe it, like what we just described. Um, that's what I did my dissertation on. That was a large portion of what I did as a graduate student. I've just never grown up from that. I still love sulfur, and probably because it plays such a key role in the ecology of microbes in Yellowstone.
Excellent. Um, Allie from California asks, "Do thermophiles compete with other thermophiles, and is there a top dog thermophile?"
So microbial ecosystems are, you should think about them as being no different than any other ecosystem. Organisms have to make a living, right? And so in hot springs, there's a finite amount of resources, right? We talked about electron donors or electron acceptors or nutrients, if you will, just blanket nutrients. There's not enough to go around, so organisms are going to compete, and they do compete. The most successful organisms are those that tend to be the most dominant organisms. And I'll point you to two different types of organisms that are the top dogs of their respective environments. So, if you've been listening to this entire podcast, you'll remember we talked about the acidic hot springs and the circumneutral to alkaline hot springs. And I tend to not study photosynthetic ecosystems. I'll just point this out. So, if you see green coloration in a hot spring, you know that it's less than 73 degrees centigrade. Okay, that's the upper temperature limit for photosynthesis. We study organisms that live above that. In circumneutral to alkaline hot springs, the top dog is generally an organism called Thermocrinus. And that's a group of organism that forms those long pinkish to white filaments in hot springs.
Oh yeah.
So you'll see those if you're down in the Old Faithful or the Middle Geyser Basin type areas. That those are classic circumneutral to alkaline type hot springs, oftentimes dominated by thermocrinus. If you're in those sulfuric acid, acidic hot spring areas, the top dogs tend to be sulfolobus type organisms. And so they're just excellent at what they do. They can outcompete other microbes that live in those hot springs and become dominant.
Got it. Interesting. Um, John from Washington asks, could a thermophile live on the sun?
Hmm.
Maybe a
Um,
little too hot, but
it
maybe.
definitely too hot. But it's an important question because it makes you think about, are there environments on Earth that do not host life? Well, the best example of that is if you walk up to a volcano like Kilauea or others, and you can see that molten magma. It's just simply too hot for microbes to live there. So if you're above 122 degrees C, that's the upper temperature limit for life, which molten magma definitely is, and the sun is even hotter than that. Millions of degrees. It's just too hot for microbial life.
Where are some locations that visitors in Yellowstone National Park can go to observe thermophiles in action and also put a disclaimer in from a distance on a boardwalk and staying on the boardwalk?
Yeah, well, that's the most important point.
Yes.
Is you definitely need to follow the rules. The rules are in place in a place like Yellowstone for your safety, and I'm not just saying that on behalf of the Park Service. I have seen what can happen to people and animals when they don't follow the rules and walk off boardwalk. My favorite place in all of Yellowstone is Norris Geyser Basin. And so Norris is one of the few places you can go to and you can get it all. You can get the acidic hot springs, you can get the alkaline hot springs. Great examples of photosynthetic ecosystems. Others that are not photosynthetic, that are living off of chemical energy. Again, those that are hotter than 73 degrees. Um, Norris is a great place to do that. But quite frankly, there is no geyser basin in Yellowstone that is not worth going and spending a day at. I love the Upper Geyser Loop Trail above Old Faithful. You'd be remiss to not go and walk the boardwalk and see Grand Prismatic, like just a phenomenal hot spring. The Sapphire Pool you can see from there too. So any of the geyser basins are great, but my personal favorite is Norris for certain.
Nice. Do thermophiles influence the incredible colors found in Grand Prismatic?
Yeah, yeah. So if you are there's a overlook for Grand Prismatic on the backside of